Today, we're going to crack open a fascinating cellular detective story. For decades, we've known about some of the biggest genetic risk factors for Alzheimer's, but exactly how they cause damage has been a bit of a mystery. Well, a recent study from Weill Cornell Medicine has basically taken on the role of lead investigator, finally connecting the dots and showing us how the brain's own guardians can turn into its worst enemies. We're going to unpack what happens when a perfect storm of genetic risk factors collides inside the brain, all based on some seriously groundbreaking new findings. All right, so let's start with the big question, the one that really drives all of this research. Why is it that some people seem to face a much higher risk for Alzheimer's than others? We all know the risk isn't spread out evenly. For some folks, the odds just seem stacked against them. Well, scientists are finally starting to piece together the clues. Okay, so every good mystery starts with its cast of characters, right? Well, in the case of late-onset Alzheimer's, we have three prime suspects that, when they get together, create a really dangerous situation. First up, we have the notorious APOE4 gene. You can think of this one as the ringleader. It is, without a doubt, the single strongest and most common genetic risk factor we know of for late-onset Alzheimer's. It just dramatically jacks up a person's risk. Next, meet the accomplice, a rare but really potent variant called TREM2-R47H. Now, this gene is only found in the brain's immune cells, and while you don't see it as often as APOE4, it still significantly multiplies the risk of getting the disease. Now, here's the thing. These genetic suspects don't just operate in a vacuum. They carry out their work at a very specific crime scene, a brain that's already dealing with something called tau pathology. This is basically the buildup of these toxic protein tangles inside brain cells, and it's this damage that directly tracks with a patient's cognitive decline. APOE4. Just having this can ramp up your risk by up to 12 times. That's huge. Second, there's a less common variant, TREM2-R47H, but it still packs a major punch, increasing risk by up to 4.5 times. And the third factor? Sex. We know that statistically, women have a higher incidence of Alzheimer's. So this leads us to the critical question, the one at the very center of this new study. What in the world happens when you put all three of these suspects together? What kind of chaos could this triple threat combo actually unleash on the brain? Well, to find out, researchers did something incredible. They engineered mouse models that had all three risk factors. They set up a pretty incredible experiment. To figure this out, the researchers got really creative. They started with mice that were already set up to develop those tau tangles that we see in Alzheimer's. Then, they used genetic engineering to give these female mice both the human APOE4 gene and that risky TREM2 variant. What they created was a triple risk model, a unique chance to see what happens when you put all the main suspects in the same room. And the evidence that came back, it was conclusive. And it was pretty alarming. The brains of these triple-risk mice just degenerated so much faster. They saw major brain atrophy. That means the fluid-filled spaces in the brain got bigger because brain tissue was literally disappearing. They also measured a huge loss of synapses, those critical connection points between neurons. And finally, they saw problems with long-term potentiation, which is the actual cellular process that forms the basis of learning and memory. So, with the damage confirmed on a brain-wide level, the investigation had to zoom in. The next logical question was, okay, but who is actually pulling the trigger here? What's the cellular mechanism that connects these genetic risks to all of this destruction? Well, the investigation pretty quickly zeroed in on one particular cell type, the microglia. You can think of these guys as the brain's dedicated security guards and cleanup crew. In a healthy brain, they are absolutely essential. They're constantly patrolling, getting rid of waste, and protecting our neurons from any threats. But in these triple-risk mice, the security guards went totally rogue. They weren't just responding to a threat, they were stuck in a state of permanent high alert. All the evidence pointed to them becoming chronically overactive and dysfunctional, which, as it turns out, is incredibly toxic to all the neurons around them. But remember, that TREM2 gene is found only in these cells, which makes them a prime suspect. So how does this happen? The researchers pinpointed a specific mechanism called the cGAS-STING pathway. The best way to think about this is like a cellular burglar alarm. Under all this stress from the genetic risks and the tau, the cell's little power plants, the mitochondria, start to leak their own DNA out into the cell. A sensor protein named cGS sees this DNA where it shouldn't be and trips the alarm. This flips on another protein, STING, which then just blasts out a powerful inflammatory signal called interferon. Now, this system is supposed to fight viruses, but here it's being triggered from the inside, putting the cell and all its neighbors on a constant damaging war footing. Okay, so you've got this cellular alarm just blaring nonstop inside the microglia. What does that kind of chronic stress do to the cell itself? Well, the result is something straight out of a horror movie. Let's go back to our security guard analogy for a second. What happens when that guard is totally overworked, completely burned out, and stuck on high alert 24/7? Well, they don't just quit. In the cellular world, they enter a really strange and really dangerous new state. This state is called cellular senescence. The cell basically becomes a zombie. It stops doing its normal job, it stops dividing, but here's the creepy part. It doesn't die. Instead, it just sits there, pumping out this toxic cocktail of inflammatory stuff that damages all the healthy cells around it. It's no longer a guardian. It's become a source of poison. And the researchers found the smoking gun for this zombie transformation. I mean, just look at this chart. It shows the levels of a key marker for senescence, a protein called P21. You can see it clear as day. In the microglia from the triple-risk mice, the levels of this zombie cell marker were just dramatically higher. There was no question. They had turned senescent. And this brings us right to the core conclusion of the whole investigation, summed up perfectly by the researchers themselves. It's not just about the genetic risks, and it's not just about the zombie cells. It's the entire chain of events. The cGAS-STING alarm system gets tripped, and that directly causes the microglia to turn into these toxic senescent cells. So the case is basically cracked. We know the suspects, we know the mechanism, and we know the awful outcome. But in science, solving one mystery is really about opening the door to the next big question, which is, what do we do about it? I mean, really, why does identifying this super specific pathway matter so much? Because for the first time, it gives us a clear, actionable target in the fight against Alzheimer's. So here it is, the whole story, the complete causal chain laid bare. It starts with the combination of those three big risk factors. That leads to cellular stress and leaky mitochondria. That in turn trips the cGAS-STING alarm. The constant alarm transforms the brain's microglia into toxic zombie cells. And finally, those zombie cells pump out harmful stuff that destroys synapses and neurons, leading to brain atrophy and ultimately cognitive decline. And this is where the potential for new treatments gets so exciting. For decades, a lot of Alzheimer's research was focused on clearing out the plaques and tangles, the symptoms of the disease. But this discovery shifts the focus entirely. What if you could develop a drug that doesn't just clean up the mess, but instead just turns off the faulty alarm? What if we could calm that cGAS-STING pathway in microglia and stop them from ever becoming zombies in the first place? And that leaves us with a really powerful final thought. This research suggests that maybe the key to treating Alzheimer's isn't some all-out assault on the damage we can see, but a much more delicate mission, restoring order to the brain's own security force and just reminding its guardians who they're supposed to be protecting.